Hello everyone, welcome back to our channel Alpha Doctors, where today we are going to continue our section on the microbiology. So the last time we studied about the bacteriology and basic bacterial structures. Today we are going to continue our session and study in detail about the staining techniques of different bacteria. So the first and foremost staining technique that's most commonly used is the gram staining technique. So the gram stain is the first line lab test in bacterial identification. So those bacteria with thick peptidoglycan layer, we already studied that the cell wall of bacteria is made up of a peptide chain and the glycan chain. So there's a polysaccharide and there's a protein layer. So the thick peptide, those bacteria having thick peptidoglycan layer, which means they can retain the crystal violet dye within themselves or within their cell walls, and these bacteria are termed as the gram-positive bacteria. So the gram-positive bacteria are those bacteria which stain which stain positive with the gram stain, and those which retain the gram stain within their thick peptidoglycan layer. Whereas those bacteria that contains the thin peptidoglycan layer, that either turn red or pink and do not retain the dye within their cell wall or cell membrane, and hence they are termed as gram-negative bacteria. So here you can see a short mnemonic for those organisms that do not gram stain well, which means they are gram negative bacteria. So these microbes may unfortunately lack real color but are everywhere. So they have different reasons for not staining or poorly staining with gram stain. For example, Treponema and Leptospirea. These organisms are too thin to be visualized. So the mycobacteria is a special type of bacteria as we will study. So mycobacteria has a cell wall that has high lipid content. So we studied that those bacteria which contains a thick peptidoglycan layer will stain positive, whereas mycobacteria do not contain peptidoglycan layer. Instead, they contain a high lipid content within their cell membrane and cell wall, for which reason they do not stain well with the gram stain. Then here the mycoplasma and ureaplasma. So the ureaplasma and mycoplasma do not contain any cell wall. That is, these organisms lack the cell wall, and there's a peptidoglycan layer is present in the cell wall. So these organisms again do not stand well with the gram staining. Then we have some organisms that are primarily intracellular, that is, they are found inside the cells, including the Legionella, Rickettsia, Chlamydia, Bartonella, Anaplasma, and Ehrlichia. So these organisms are primarily reside within the inside the cells of the host cytoplasm. Therefore, these do not stand well with the gram stain because gram stain cannot basically stain the host cells. And chlamydia is a special type of organism because its peptidoglycan layer contains less number of the methamic acid. So methamic acid presence is another important factor to determine the type of staining. Then Giemsa stain. So Giemsa stain is reserved for few specific organisms such as Chlamydia, Rickettsia, Trypanosomas, Borrelia, Helicobacter pylori, and Plasmodium species. So out of them, you need to the organisms that you need not to be forget are Helicobacter pylori and Plasmodium. So Plasmodium and Helicobacter pylori are the important organisms that stand with each stand, and you will be tested again and again on your world for this. Then we have the PAS staining, that is the periodic acid shift stain, which stains basically glycogen mucopolysaccharides. So these are used for the diagnosis of Whipple's disease. So it's an important and easy way to remember that PAS positive is a stain that is mainly used for the Whipple's disease. And because why does it stains the Whipple's disease? Because Whipple's contains glycogen in its cell wall, and PAS is useful for staining glycogen. Then you have the Zeal Nielsen stain, which is also known as the carbonyl fusion stain, which stains the acid fast bacteria. So in acid fast we studied excuse me. So in acid fast we studied that mycobacteria is an organism which cell wall is made up of lipid content. So here Zeal-Nielsen stain is an important stain which stains the mycolic acid in the cell wall that is made up of lipids, and it stains the mycobacteria. And there is another organism that is known as the nocardia. So nocardia is also a bacterial species which contains mycolic acid in its cell wall, and it is also stained with Zeal-Nielsen stain. Likewise, we have one protozoa. So this protozoa, that is the Cryptosporidium. And Cryptosporidium is an important organism uh, that contains mycolic acid in its cell wall, and it causes disease. And those that are infected with AIDS, we will study about it in detail in our subsequent sections. Then we have the Indian ink staining. So the Indian ink staining is useful to stain the organisms such as Cryptococcus neoformans. So Indian scan is useful to stain the lucicermine, that is an important structure within the cell wall of certain bacteria such as Cryptosporidium neoformans. So we have two species. One of them is the Sporidium oocyst, which is stained with Zee-Nielsen stain. Then you have the Cryptococcus neoformans, that is stained by the Indian ink stain. And the difference between them and the pathogenesis and disease-causing ability will be studied in our next sessions. We will detail about these protozoa in detail. Then you have the silver staining. Silver staining is useful for fungi such as coccidioides, Nemocystitis, Jirovecii, Legionella, and H. pylori. And then we have the fluorescent antibody stain. Fluorescent antibody stain can detect many bacteria and viruses, including Nemocystitis, Jirovecii, Giardiasis, and Cryptosporidium. So, one important bacteria that you need to remember with the fluorescent or the FDA-ABS is the syphilis. Syphilis is one of the most commonly tested organisms that is stained with FDA, that is the fluorescent antibody stain, and this is tested again and again. So overall, this was a brief overview of the staining. First of all, we studied the gram staining. Those organisms that stain positive with the gram stain are known as gram-positive organisms, whereas those which do not stain well with gram stain are known as the gram-negative staining organisms or the gram-negative organisms. The primary the primary reasons for organisms not staining with the gram stain could be because they lack the cell wall, because the cell wall is not thick enough, or because they lack important um, because they reside inside the cells, or because the cell wall is made up of mycolic acid or lipid content such as mycobacteria. Then we studied the past staining, which is Whipple's disease. Then we studied about the Zeel-Nielsen stain, which is 10 organisms, that is the mycobacteria, the nocardia, and the Cryptosporidium species. Then we studied the Indian ink stain, which stains the Cryptosporidium or Cryptococcus neoformans. Then we studied the silver stain, which stains certain fungi, Nemocystitis, and Legionella and H. pylori. And then we studied the FDA stain, which stands for syphilis. In our next sessions, we are going to study about the properties of the growth media, such as and special culture requirements such as the chocolate agar and the Thai-Martin agar, which are useful for the growth of different organisms and are most commonly used in culturing of the organisms and growing them artificially. If you have any questions, you can ask in the comment section. Hope you understood the gist of this video. Thank you.